How might a relatively small island, like Texel, be affected by these microplastics? And more specifically, what role do pumping stations play in this? These stations often transfer to land-based water, sometimes containing litter, into surrounding water bodies and eventually the sea. So what happens when this litter reaches filter-feeding organisms like oysters? These creatures filter large volumes of water to feed and breathe, meaning they're especially vulnerable to ingesting microplastics. This can affect their health, their growth, and even the quality of seafood we consume.
On Texel, we also visited pumping station Eierland in the Koksdorp. To prevent fields from flooding, this pumping station pumps exit groundwater from land out to sea.
I'm standing in the outlet of a pumping station right now, and as you can see, there's a bunch of oysters living here. And oysters are actually filter feeders, which means that they have a specific filtering system which they use to take up organic material, which they use as food. However, there's a catch. The food particles are basically the same size as microplastics, and therefore they also filter the microplastics out of the water. Unfortunately, this also means that oysters now contain microplastics.
Our hypothesis is that the vicinity of pumping stations have a positive effect on the amount of microplastics in filter feeders. In this research, we will focus on the Pacific oyster, an abundant oyster species in this area.
To sample this, we're in it right now at a pumping station on Texel in the north. And we're going to measure the amount of microplastics in different locations. So over here, we're going to take 3 samples in the north. Another group is taking extra samples more to the south, where it's more further away from a pumping station.
At the locations with a pumping station, we made a gradient from directly next to the outlet of the pumping station to a distance of up to 2 kilometers from the outlet of the pumping station. At each location, we made 3 sublocations, and at each sublocation, 3 samples will be taken, each sample consisting of 3 oysters. This means in total, 9 oysters will be taken per sublocation, and 27 oysters will be taken per location. Since the muck bay is not large enough to make a gradient, random samples from the muck bay have been taken, and these are seen as one sublocation.
In our field lab, our oysters undergo a purification treatment to be able to expel up to 40% of their microplastic. You can see this as a detox program for oysters. This water will later be sampled for microplastics. In order to have a triangulation of methods, we also dissolve some oyster samples in potassium hydroxide to analyze the microplastic content in the oyster meat. To do this, the oyster meat was measured before dissolution.
From the stereoscope analysis, we identified microplastics as being brightly colored strings or fragments. We counted the total number of microplastics in a petri dish and distinguished between the color and shape. The most common find was blue string, which was identified as polyisobutylene. Red string was also identified as PIB, and white fragments were shown to be polyacrylamide.
With this method, we have found the following results. At the Koksdorp, we found less microplastics than in the control group, which was the Mokbai. The data from the Koksdorp showed no significant connection between the amount of microplastics and the distance from the pumping station. At Ouderschild, we found more microplastics than in the control group, with the highest amount of microplastics right next to the pumping station and the least amount at the furthest location. The microplastics found at the furthest location were also comparable in numbers to our control group. This leads us to a gradient that can be statistically tested with a linear regression test. From this test, we got a p-value of 0.02, which is lower than our alpha of 0.05. So we can conclude the pumping station does have a positive effect on the amount of microplastics in the oysters. Regarding these results, we do have a discussion point, since we found more microplastics when a lot of people were walking past us and our samples. To test the amount of contamination, we took random samples of the air and surfaces in the lab and analyzed these samples on the amount and the type of microplastics in the contamination, so we could identify a part of the contamination when analyzing our samples. To prevent contamination and make it easier to identify contamination from our clothing, we wore lab coats to make all of our contamination white, and we also put a lid on the samples as much as possible.
With all of this said, our conclusion still stands. So smile away, boys, and thank you for watching, and until next time. Bye bye!